Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to today's webinar titled Integrating Patient Engagement and Trainee Development in Preclinical Research, featuring Dr. Christopher Perry, Associate Professor in the School of Kinesiology and Health Science at York University, and Dr. Homira Osman, Vice President of Research and Public Policy at Muscular Dystrophy Canada. Today, they will come together to discuss how integrating clinical, industrial, and not-for-profit partners with preclinical research labs offers great potential to improve research impact in a manner that focuses questions to the unmet needs of people living with muscle disorders. I also want to take this moment to thank our partners at the American Physiological Society and the sponsor for this event, Aurora Scientific, for making it all possible. Without any further delay, I'm going to welcome Dr. Perry. Hi, Chris.
Hello.
Please take it away.
Okay, thank you very much. I'd like to thank you, Sydney, for having us here, as well as the American Physiological Society, Aurora for sponsoring, and Muscular Dystrophy Canada. This is a great opportunity for me as a lab director to be part of what I think is a pretty important conversation. I'd like to start by giving an overview of what our research is to help sort of contextualize why I think it's going to be important for us as preclinical researchers to be more engaging with people living with the diseases that we study, and through that process, putting our trainees front and center in speaking with people with these diseases, as well as all of our partners, clinical partners, industry partners, not for profit. I think the trainees need to be front and center, and like to have a discussion about why that is. So I've put some images on the right side to give an idea of what it is we study. We study diseases that cause muscle weakness. Very often that's caused by muscle wasting, but actually not always. And we focus on metabolic dysfunction. That's why I threw up this mitochondria picture, which I'll talk about next. Of course, first, the most important slide, I'd like to highlight the students in our lab. Past and present, and they've varied in what they are studying, what their thesis research was focusing on, from Duchenne muscular dystrophy, cancer metabolism, and we've got models of colorectal and ovarian cancer-induced muscle weakness, autoimmune myositis, and through collaborations, type 1 diabetes. And so we don't define ourselves by a particular disease. Rather, we're interested in the basic science, the metabolic contributions to muscle weakness and muscle dysfunction that allows us to study multiple diseases. And we're very, very, very appreciative and would not be where we are in our research without collaborators across the world. And of course, our funding partners, including Muscular Dystrophy Canada. So our research program, as I mentioned, we look at the metabolic dysfunction in muscle weakness disorders. And we can call that a metabolic stress. So to what degree are people having a tough time getting out of bed, walking, breathing, pumping blood with their heart due to actually a metabolic problem inside of their muscles? And that brings us to a part of muscle cells called mitochondria, these tiny little structures inside of muscle cells, and then indeed in many different cell types, not just muscle. And they're a major source of energy, where they take the sugar and fats from our food that we eat, and they essentially release that energy from our food. And that energy goes towards making a muscle fiber contract. And that's true for your arms, your legs, so limb muscles. That's important for mobility, your ability to move. That's also true within the muscle cells of heart. And that obviously is critical for your body's ability to pump blood. It's also true within muscle fibers of the diaphragm that is important for helping us breathe. So by understanding how muscle cells produce energy and asking a question, to what degree do muscle weakness disorders arise in part from disruptions in metabolism? And, to what degree does that metabolic dysfunction contribute to immobility, heart problems, and breathing problems? Mitochondria do a lot more than just produce energy. They can also release stress signals, things I won't get into today, but things like reactive oxygen species, other signals that can really stress muscle, sometimes in a good way and sometimes in a bad way. And when mitochondria really start to get stressed themselves, they can actually cause cells to die, which is an intriguing line of research to understand how metabolism can actually lead to atrophy. So that's a high level overview of what we do. I'm going to show you some examples of our research and how we're using that to now modify our approach to research with patient engagement. But first, our research. So while I mentioned we look at a number of muscle weakness disorders, a major area of research for us is Duchenne muscular dystrophy. And this is a genetic disease that occurs primarily in boys. The statistics and prevalence varies depending on what you read. It's a ballpark around 1 in 5,000 live births, predominantly in males. And by childhood, parents may start to notice some differences in posture and mobility. And depending on the severity of the disease and the speed of its progression, by teens, a boy could be wheelchair bound. And by adulthood, there's progressive muscle loss and more weakness. Eventually, therapy may be required to go on a mechanical ventilator to assist with breathing, heart medications to help boost heart function, and strong anti-inflammatories, glucocorticoids, to try to slow the degree of muscle wasting. And it can become particularly problematic because as muscle fibers waste away, they can be replaced by scars. So the muscle builds up scars, and that is thought to contribute to the progressive muscle weakness. And so it's a disease of muscle atrophy, muscle weakness, not just in the limbs, but also the heart and the diaphragm and breathing muscles. And so this decrease in functional muscle results in muscle weakness. And that's a very important term because not only are muscles decreasing in mass, but what muscle remains can become less functional. Even the fibers that remain themselves can be weak. So it's not just weakness because of smaller muscles, it's weakness because of something wrong within the fibers that remain. So decrease in functional muscle as well. And so the leading therapy is corticosteroids. It started with prednisone. Some clinics still prescribe prednisone. There's other forms of corticosteroids now, like prednisolone and deflazacort and so on. But it's basically treated by targeting inflammation, because you can imagine with so much muscle damage occurring and muscle wasting, the body is really ramping up its defense mechanisms to try to assist with repairing. And inflammation is a good thing. It helps with healing. But the problem is when there's so many muscle fibers that are essentially atrophying, there can be an excessive inflammatory response, too much inflammation. So perhaps somewhat ironically, while inflammation is needed for treatment, needed for healing, the leading treatment is to lower the inflammation. So you're not getting rid of it, you're just bringing it down to a level that helps you heal. And it's proven to improve muscle strength, increase the number of years that someone is mobile. There's some clinical evidence that nutritional therapies, there's a study or two that showed creatine supplementation was associated with improved muscle strength. And that's about it. Now, I could very easily update this slide with recent federally approved therapies. It's more in the US and Europe for emerging gene therapies that are showing some promise as well. So that being said, there's a clear need to keep developing new additional therapies. And while a cure must be developed, in the meantime, there's an opportunity to identify other secondary contributors. What I mean by that is if inflammation is a contributor to this disease, are there other cellular problems or physiological problems also occurring that we wouldn't say is the cause of the disease, but are contributing to the muscle weakness? And that's where we come in, and many other labs as well, looking at the metabolism of the muscle cells. Looks like the metabolism is pretty messed up. And we and others are trying to figure out, is that purely associative, or is the metabolic dysfunction just yet another reason why people are having a tough time breathing, pumping blood, and getting out of the chair? That brings us to mitochondria. And so I'm going to show you some highlights from 3 different drug studies we've done. Our approach is to partner with industry to test their novel compounds that are known to boost metabolism inside mitochondria. And we test these compounds in preclinical models. And what we mean by that is mouse or cell culture models. And so here's some data from a mouse model with Duchenne. And pardon the technical terms here. What we have here, I'll start, is we look at limb muscle and we look at diaphragm. So in our studies, we always try to look at different aspects of the disease. And this is a nice sort of prelude to the topic of learning from patients lived experiences. Now, we didn't talk to any patients before doing this study. It's what we want to do moving forward. But we certainly understood from the literature, the scientific literature, as I mentioned a few times, there's a limb weakness, so mobility problems, and a breathing problem and heart problems. Now, in this study, we looked at a limb muscle and a diaphragm muscle. And before you look at all the curves and stats, don't worry about that. I just want to highlight that our approach is to try to understand the, to the degree we're trying to make our preclinical research as translationally relevant as possible, as can be done to the human condition. So if people are having a tough time moving and walking, let's pick a muscle that helps people move. So that's the quadriceps. This is from the thigh. And again, this is from a mouse. But people are having a tough time breathing, then we should also look at the diaphragm. Pretty simple approach as an example. We have some data in heart. I'm just not showing it today. Now, what we can do is actually electrically stimulate the muscle to contract. And I know you're having to use your imagination here, but we do insert a little electrical probe into the quadricep. And we're trying to model how the brain sends electrical signals to the muscle, but we're doing it artificially. And this is where Aurora Scientific's equipment comes in big time. We use their equipment to generate this data. So as we increase the electrical stimulation, and don't worry what the technical terms mean, left to right means we're just sending more electrical signals. WT means a wild type mouse. I know that's technical, but what it means is just a healthy, normal mouse. And you can see force production goes up as you electrically stimulate. It's kind of how the brain works as well. It sends more and more electrical signals to increase muscle contraction. And that's a nice, healthy curve. When you look at a mouse model of DMD, Duchenne muscular dystrophy, the blue squares, clearly it's not producing as much force. Now, we tried this mitochondrial enhancing drug from our partner Stealth Biotherapeutics that's known to get in mitochondria and boost its metabolism. And we found that mice with Duchenne muscular dystrophy that we treated with the drug for 3 weeks didn't really show any improvement of force in the quadricep. And so don't worry about the stat symbols. It's just saying both of these lines are weaker than the healthy mouse. So both mice had weakness. But what was interesting was when we looked at the diaphragm, after 3 weeks of treatment in mice, the green line was higher than the blue line. And that's what this dollar sign means. The green is higher than the blue. So this is about a 25% higher force production in the drug-treated mice. So a mitochondrial enhancing drug, by boosting mitochondrial metabolism, actually prevented some of the weakness in the diaphragm. And that's profound because breathing dysfunction is a very critical problem in people with Duchenne. And this was also in only 3 weeks of treatment. And they were in very young mice. We started somewhere between day 4 and 7 of age, like right after they're born, and only went up to 4 weeks of age. So clearly our future research wants to look at the effects of longer term treatment. Can we get that green line even higher with metabolic enhancing therapy? And by no means do we think this will ever cure the disease, because it's not a primary metabolic dysfunction is not a primary cause, but it starts to raise questions of the degree to which drugs that boost the metabolism of muscle might be added to existing therapeutic paradigms. Like, should we be taking anti-inflammatory and a mitochondrial enhancing drug? I'm not proposing that based on the single mouse study, but I just want to allude to the value of doing the preclinical research because it raises these questions. And this was done by Sophia Ramos and Megan Hughes, very, very smart students. And well, they're doctors now, and they're moving on to successful careers. But we learned that boosting mitochondria may not benefit all muscles. And that's another important thing to consider. And this is where engaging with patient partners and especially clinical partners and industry partners to understand what part of the lived experience might benefit from this type of therapy. It may not benefit all types, all aspects of the disease. Maybe it's more relevant to muscles that are always working, like the diaphragm. I'm purely speculating. I'm not proposing that. But it's important to recognize where a drug is working and where it is not working, to put your thinking cap on to say, where could this fit in future therapeutic paradigms? We tested another drug, Catherine Bellissimo. She's now a doctor as well, graduated and doing a great postdoc at Toronto General Hospital. And she tested another drug called olesazine that goes in the mitochondria and also boosts its metabolism. And here's another example of a diaphragm protocol. Now, this one's a little different, just to give you a little bit of science. What you don't see before all these lines is we actually contracted the diaphragm a lot until it fatigued. And then what we do is it's like, imagine exercising your muscles until you just can't move anymore, and then taking a 5-minute break, and then trying to exercise again and seeing how much you can come back. So after 5 minutes after fatiguing a diaphragm, we electrically stimulated it. And you can see in a healthy mouse, it actually recovers very quickly, almost 100% of its force after 5 minutes. But a mouse with Duchenne, not so good. It can't fully recover its force 5 minutes after being fatigued, so 80%. But with the olesazine treatment in a mouse, again, about 3 weeks or so, the green line's higher than the black line. So the drug allowed the diaphragm to recover its ability to contract faster after fatigue, and that's very relevant to the problem of the disease. And that was true no matter where we looked. And then if you look at muscle damage markers in the blood, it's actually called serum creatine kinase. Healthy mouse, very little muscle damage. Mouse with model of Duchenne, very high damage. And the drug actually prevented some of the damage. And that's important because the disease is a disease of muscle tearing. And so this suggests the muscle damage was prevented. But you don't, when you're careful with your experimental design and you pick more than one way to assess muscle function, you see how complex biology is. When we looked at the ability of a mouse to voluntarily run on a running wheel, a healthy mouse can run several kilometers a day, but a mouse model of Duchenne barely runs at all each day, and the drug didn't change that. So we don't know if that's because this improved fatigue, recovery from fatigue was even relevant, or translated, I should say, to improve mobility. But it's a bit tricky because voluntary running wheels, maybe the mouse just didn't want to run. It's also a behavior test. And so it's not that it's a bad experiment, it's that this is the value of adding multiple experiments, is when you see where a drug works, you see where it doesn't work, it makes you think about the constraints of the methodology guiding your interpretation and how you might use this to progress to clinical trials. It's okay if a drug doesn't work everywhere. In fact, no drug does. And we just have to capture that so we learn. Same thing, we can measure the grip strength of a mouse, and it didn't really rescue grip strength. It was down in the mouse with model Duchenne, didn't really rescue with the drug. And so that's what we need to ask ourselves. What is this data telling us? We have to recognize the mice may have chosen not to do this. So maybe the mice just feel terrible. The drug doesn't stop them from feeling terrible. But when we isolate a muscle and we remove behavior from it and electrically stimulate, we get it. We get the drug effect. So this is the value of doing multiple different assays, is to see where things work and don't work and put your thinking cap on and how to further develop a drug before going to clinical trials. Last bit of data here is with our PhD student Shivan Gandhi, and he partnered with a list of pharmaceuticals. Now, this drug is not a mitochondrial targeting drug, AOY688. It's actually a novel anti-inflammatory, and it's structured similarly to a natural hormone in our bodies called adiponectin, and it binds the adiponectin receptor and turns it on. And adiponectin receptors do a lot of things. They do enhance metabolism for more mechanisms than just mitochondria, but they're shown to be anti-inflammatory. Now that's intriguing because the current standard of care is to give glucocorticoids as anti-inflammatories. So we're interested in whether this could be a different type of anti-inflammatory. Shiv looked at the heart. So we took a cross section of the heart, and this is the left ventricle, nice and thick. This is the right ventricle, and here are the two atria. That's a healthy mouse. Now, Shiv added a stain for collagen. So we're looking at scar tissue formation. And the stain for collagen shows up as pink. And look at all this pink in the mouse model of Duchenne. It was mostly in the right ventricle, all the scars building up here. And you can't really see it. You have to zoom in on the microscope, but we saw more sort of a darker shade of pink everywhere here. Not quite as robust as down here, but more darker shade of pink being picked up by the software here, then in here. So this chamber, the left atria, and this chamber, the right ventricle, we're getting pretty scarred. And when you look at the low dose of the drug after 3 weeks or so, and the high dose of the drug, the right ventricle, actually, we couldn't find any scar tissue formation. And that's quite profound because there is no therapy approved that, to our knowledge, that can prevent scar tissue in the heart. So that's how we go about this. We partner with industry pharmaceutical partners who have novel compounds that we can test in the mouse to try to inform further drug development, with our ultimate goal of trying to get something to a clinical trial. And that brings me to the last couple slides here. Well, how do you do that? Well, our approach is to read the literature, scientific literature, and develop a hypothesis. But I think what we're learning is that the literature doesn't capture everything that we should learn about the disease. We do not have muscular dystrophy. We cannot draw on our own experience. So I'm left wondering more and more, what are we missing? Yeah, I know to measure muscle force. I know to measure muscle wasting. I know to measure muscle fibrosis. Those are the three big problems in Duchenne. But maybe there's other aspects of the disease beyond weakness, atrophy, and scar tissue that would be meaningful to the patient. So we've learned to now speak with clinicians, talk to industry partners who are thinking the same way, particularly when they're designing clinical trials, talk to agencies like Muscular Dystrophy Canada, who are the ones engaging people with lived experiences. And this is what's so exciting to work with Homira Osman and Muscular Dystrophy Canada is that's our opportunity to find initiatives and activities that we can be a part of to meet people with the very disease we're studying. And we need to listen and learn from them. And I believe in that process. I will drop the word believe. I know that the process must involve trainee involvement. It can't just be me talking with everyone. Certainly that's for building the relationships, but the trainees must be front and center. And that's what we've done so far with clinicians and industry partners, is bring PhD students, master students along, put them in leadership positions of managing these projects and engaging directly with our partners, because then they see the process as well, and I learn with them. And so as the last two slides here to transition to Dr. Homira Osman, this is a new approach in our lab, is patient engagement when we are not clinical researchers, right? Why would a mouse researcher, I don't want to use, we also study humans in other regards, but if we're studying a mouse model of Duchenne, why would we talk to a person? Well, this is exactly what I think we need to be doing. So we are becoming more active in participating in Muscular Dystrophy Canada events like the Walk and Roll Muscular Dystrophy Canada. And really my point is not to highlight what we're doing. My point is, how do we even get the opportunity to meet people with Duchenne muscular dystrophy? We benefit as researchers by the activities being conducted by Muscular Dystrophy Canada. If we participate in these, it is an avenue to learn from people with diseases that we're studying, and to learn from Muscular Dystrophy Canada professionals themselves. So we want to engage communities with neuromuscular disease. Muscular Dystrophy Canada has been wonderful in suggesting we could prepare presentations to communities about our research and start to engage in discussion and hopefully listen to their stories as well. And the trainees have to be front and center, because this, if I was a trainee, I would think, and I got to meet someone with the disease I'm studying, I would not only be inspired, I think I would really have my eyes opened. And we need to listen and learn. What are their day-to-day realities that are not available in the literature? Very brief example, I just learned muscle pain is common in women who carry dystrophin mutations that haven't manifested to Duchenne muscular dystrophy. I learned that talking to a clinician 2 weeks ago. I didn't know that. I couldn't find that in the literature. Should we be measuring muscle pain in our studies now? I never thought of that. I'm not saying we will, but that's how we can benefit, is learning other aspects of the disease, makes us reflect. And so to close, I think I'd like to learn more as a researcher on how to engage patients through structured systems. And I've taken a great interest in this framework that's been developed. And it was actually sent to me by my father-in-law, who doesn't have muscular dystrophy. He has another disease, and he's a patient partner in clinical research. I'm actually learning from him quite a bit about how in clinical research he's being involved to inform researchers of the lived experience. And that really got me thinking, why aren't we doing that with our mouse studies? And so I couldn't find any framework for engaging patients for preclinical research. And I had a lovely conversation with Dr. Homira Osman. I'm looking forward to her presentation next. Because I think there's great opportunity to learn how they help researchers, clinical researchers, engage patients, and to learn from them. Maybe we can model that as preclinical researchers to go through similar structured programs through Muscular Dystrophy Canada and other agencies as well down the road to learn how to approach a person with the disease, learn how to listen and to think more carefully about how we can improve our research at a preclinical level. And with that, I'll very much looking forward to Dr. Homira Osman's talk next.
Thank you very much, Dr. Perry. It is really a privilege to be here and included in today's conversation. I am the VP of Research and Public Policy at Muscular Dystrophy Canada, and today I will just, continue that presentation by talking about really moving patient-oriented research forward. Before I do that, just for those who aren't familiar on today's call, a little bit about Muscular Dystrophy Canada. So MDC is a national organization. We represent Canadians affected by neuromuscular conditions. Our focus in terms of our organization is to fund research, conduct research as well, and also support with the here and now. So what does that mean? We support individuals with programs, services, funding for equipment, assisted devices, technologies. We also are there really to walk hand in hand and shoulder to shoulder with those who have neuromuscular conditions and their family members as they navigate the complex medical and community systems. We also support advocacy, and so that could be individual advocacy at a local level, provincial, and at a national level, like the current rare disease strategy. At Muscular Dystrophy Canada, we represent hundreds of neuromuscular conditions. While our name is Muscular Dystrophy Canada, and as Dr. Perry spoke about Duchenne muscular dystrophy, that is certainly one of the conditions that we cover, but we do cover hundreds. And we often talk about them in terms of they can be individually rare conditions, but collectively common. These can be conditions that are hereditary or immune-mediated conditions, and they typically affect the muscles and contribute to weakness. We also cover conditions that impact the neuromuscular junction or peripheral nerves. I really want to focus our conversation about this principle called nothing about us without us. I'll first talk about how we at MDC adopt that principle and then really speak about it in terms of research. At MDC, at all levels of how Muscular Dystrophy Canada runs, at all levels of governance and how we run our programs and services and perform our mission, we include people with neuromuscular disorders. These could be parents of children or young adults who have neuromuscular conditions, or individuals themselves who have neuromuscular conditions. These individuals serve on our board or advisory committee or work at MDC as directors or in other roles as well. It's really important to really be grounded in that lived experience for an organization that represents Canadians affected by these neuromuscular disorders. Now, nothing about us without us is a phrase, and it typically is that idea that services and policies should involve the engagement and participation of members and groups affected by that policy. It has been a phrase that's been used traditionally as part of the disability and activist movement. In recent years, it's now being used more so in the context around research. Nothing about us without us is often now used in terms of we should not be researching certain populations without their input. And it's not just about input, and we'll talk about that today. It's not just about input, it's about meaningful involvement. And so really there is now emerging research that actually says that research studies and research teams should really focus on including people, patients, and their family members in research to really make sure that the findings are transferable, meaningful, and can be validated. We've all probably heard this term bench to bedside. And oftentimes when you're thinking about that, this image might come to mind, this idea that basic science research is at T0, is at that first level, and then eventually it moves into humans and clinical trials potentially, then it moves into patients, it's then picked up in clinical practice, and then it goes into the community. As I'm walking through that, you're likely thinking that could take quite a bit of time, and it certainly does. And it seems almost linear as well. Although, many of these models, including this translation process that I'm showing right now, shows the relationship to be bidirectional. Perhaps it's better to view it in this format. And I apologize, this is in French, but I could not pass on how beautiful it really shows where we can re-envision this bench to bedside model. So what you can see in the green is really thinking about, are there opportunities to engage in translational research where it starts from the patients first? And we have seen some examples of that, and most recently even in muscle stem cells of those with Duchenne muscular dystrophy, where stem cells that were part of a biobank in Montreal were then studied and tested using certain medications that were being tested. The same could apply with images, MRI scans, CT scans, X-rays, DNA samples, and then that moves its way back into that basic science research laboratory. Or the other way around, where at the basic lab level, where it starts in animal models, like in the mouse model that Dr. Perry described, and then eventually moves towards patients. Now, these two examples that I spoke about today around bench to bedside, bedside to bench, and this translational process is really referring to patients being involved in research in the context of more so as a participant or being offering their information. However, patient engagement in research really refers to a meaningful collaboration. And Dr. Perry started to talk about his father-in-law being a patient partner. So I want to speak about that. Really, when we talk about patient partners or patient and parent partners or patient and family partners in research, it's really about working on research projects in a collaborative way, in co-creating research. And so when we talk about patient engagement in research, oftentimes this has really been well documented in clinical research, but not as much in preclinical laboratory research. It's less adopted. There is, these are the levels of patient engagement in research. And so, when we're moving away now from inform, participate, and consult more towards that involve, collaborate, having patient and parent partners lead and support. So there was a study that I want to bring up here where they did look at patient engagement in preclinical laboratory research. And, first of all, very limited in terms of this type of information, but frequently the studies were ones where patients provided education or patients were involved in priority setting. So there is the James Lind Alliance priority setting partnerships where basic science researchers or preclinical researchers can engage in, and they can really work with the patient community to better understand what are those questions, what are those parts of the experience in the disease that are unanswered, that are important to answer. And that then can inform research questions at the preclinical science level. And really, many of the researchers that have included patient partners really talked about it was a mutual learning opportunity. There were barriers, however, reported, such as differences in knowledge and research experience, which can challenge communication and limit meaningful collaboration. So with that said, I wanted to highlight that there are two recent training programs. One is by the Canadian Institutes of Health Research, CIHR, the Institute of Musculoskeletal Health and Arthritis, IMHA. They've developed online, self-directed, free modules, and they have a series of modules on what is patient engagement, what does that research process look like, what does it look like to set up a research project? And also for preclinical research scientists, how is it to engage with patients and families? What are some good tools and resources? At MDC, we've also started, together with the Neuromuscular Disease Network for Canada, our own set of training modules for patients, parents, and also researchers called Import ND, which can be accessed at importnd.neuromuscularnetwork.ca. This is very similar to the IMHA ones. However, it's more so focused on inclusion, diversity, equity, accessibility, and indigeneity. It also is talking more about neuromuscular specific examples and case studies. So while patients and parents have expertise and lived expertise in their disease, we know that it can be challenging sometimes to participate in research on those teams. This type of training will support that. And same for researchers, for preclinical researchers, while they become disease experts in a certain area and become muscle disease experts, for instance, they might not have ever met a patient or have ever been part of the patient community. So opportunities like this to train are really helpful so that they can meaningfully engage in a way that is respectful and in a way that will really mutually contribute to positive outcomes. With that, I am looking forward to today's discussion and welcome any questions as well.
All right. Thanks so much. Really great presentations, both of you. Did you want to move into the discussion portion?
Sounds good.
Okay, I'll let you guys carry on.
Yeah, actually, I wasn't aware of some of those training modules, both at CHR and ND Canada. So I've already started Googling as you were talking. Certainly, I think is a there's 7 things I want to say at once. I think certainly go to that CHR website and have a look at IMHA. But this Import ND, is that something that researchers just email Muscular Dystrophy Canada and ask to learn more? Or is there a website available? Or?
Yeah, for the Import ND, there is a website available as well, and I'm happy to probably put it in the chat for those. And so actually, it's a few components in terms of that. It's a self-paced program. There are 3 modules as part of Import ND. Upon completion, researchers or patients, family members receive a certificate, but then there's this next step to it. You're then entered into a repository, should you wish, and if a researcher like yourself says, hey, I am studying this mitochondrial disease, are there any patients who've completed this program in the Toronto area who also have this mitochondrial disease or who have Duchenne muscular dystrophy? Can you match me with them? Then MDC can help to support with that matching as well. And then also through, as I mentioned at MDC, we fund research. We also fund partnerships like this. So we really believe that when patients and family members get involved in these projects, that they should be compensated and that they should be engaged early on. And sometimes without a research grant funded, it's hard to include a patient partner. So some of these funds help to support that so that you are meaningfully including a patient partner, even at the time of idea conception or writing up a grant and looking at the methodology or expected outcomes as well.
Wow, that's highly structured.
Yeah.
I'm not aware of any other agencies that have even advertised for different diseases this opportunity. Of course, I don't study every disease, but even the other muscle weakness disorders we're looking at, I pay attention to their sort of the parallel to Muscular Dystrophy Canada, different cancer societies and so on. I've never heard of this. I think that's way ahead of the curve.
Absolutely, and we're really proud about having this training program, but also really making sure that it's sustainable and really making sure that it's not just information, right? So oftentimes we all take, log on to webinars or take information and training, but really moving it into action is so important to us. And I think that's what we would love is for more researchers, preclinical, clinical, to include patient and family members in their research teams. And through that, it will be a learning process. We'll learn about what some challenges and barriers are that we can then address as well.
Right. Yeah. I've had chats with colleagues about, what if we're doing a cell culture study or a mouse study, what could we possibly learn that would be relevant to changing our view? Because very often they're thinking of cellular mechanisms, this particular enzyme, this particular gene, and it's clear from the literature what the rationale is to study that particular molecular contributor to the disease, and therefore even a clinical input wouldn't change that perspective. But I think where personally I think where the input really becomes valuable is obviously we're not looking for someone to tell us what particular gene to study. We know that. It's how to design the study as a physiologist to look at the primary outcome in the mouse model or the rodent model of the clinically relevant measures of the disease, the weakness, the atrophy, et cetera, in the case of neuromuscular diseases. And even that we understand from the literature. We know what to do there. But our experience has been so far that it's about other outcome measures that we're not aware of, other outcome measures we should be thinking of. You know, what if we talk to somebody with a neuromuscular disease and they say, you know, I'm just picking something random here. By year 5, my knee started to hurt. And we go, oh, well, that's odd. Is that a unique situation? Well, maybe now I have a question in mind. I could go to our clinical partners and say, I heard this. Do you see that in all your patients or some? And for all we know, they may say, oh, that's pretty common. I didn't know that in the literature. Now, knee pain I haven't heard about, but in the case of, I mentioned, we learned actually from a clinician at the neuromuscular disease meeting you and I were at 2 weeks ago, that women who do not have Duchenne but are carriers of the dystrophin mutation tend to have, many of them have muscle pain. So I went to the literature, and maybe I've missed a study. I didn't know that. So maybe we should be measuring muscle pain. So I personally think if I attended your module and got to speak with partners, it's other primary outcome measures we should be thinking of, not just the mechanism.
Yeah, I absolutely agree. And, you know, as you were speaking, Dr. Perry, what I was also thinking about is some of these conditions are rare, and like Duchenne muscular dystrophy, where it might not always we have these experiences, the patient experiences well documented in the literature, but then even more so to have investments in research to focus in on the carriers and the carriers, female carriers experience. Then you're looking at a rare disease, and then now you're looking at an even rarer population and that those experiences are not well documented, still important, but not well documented. And so that's why some of these questions, even at the preclinical level, are not really focused on it. Same with, I have seen several examples where preclinical researchers have met with patients and shifted the outcome measures or how they're really measuring or capturing muscle weakness, for example, as well.
Right. So the registry that Muscular Dystrophy Canada has is fairly broad then. Is that right? Like not just so different subtypes of diseases, or maybe that's not the right word. Do you have a registry of carriers as well?
That's a good question. So at MDC, we don't necessarily have a patient registry. We support the Canadian Neuromuscular Disease Registry. It's called CNDR. At MDC, we do, however, have a data hub. So when someone comes to our organization who has a neuromuscular condition or a family member, they register, and that registration is confirmed by a healthcare professional. In that case, both carriers and persons who are affected by Duchenne are included in our data hub. And so they hear about such opportunities, such training opportunities or research studies, and they get involved in that way.
I see. Have any of them ever been, or have you been involved in setting up keynote speakers, patient partners at conferences?
Absolutely. So again, you know, something that I mentioned earlier is patients and family members really they become so sophisticated and really knowledgeable in their disease area, and they really launch into their journey in advocating for themselves. Having said that, participating in research or giving keynotes are not innate skills for everyone, and so we really help to support patients and family members to get involved in such opportunities and become prepared. And so so when I'm talking about research, you know, we're talking in the context of clinical and preclinical, but I think an important element of research is knowledge translation or knowledge mobilization and science communication. And so in this scenario where a patient is able to, in a very well-versed way, share their story is something that we also include, because we really believe that science communication, knowledge translation, sharing your story, storytelling is incredibly important in talking about that patient lived experience.
Right. We left a mitochondrial disease meeting once. I think it was 2017, and we were that researcher in the audience listening to someone on stage who had a disease and shared it. And the students and I walked away saying, the research talks were fantastic, but that one was the most inspiring, and we all wanted to get back to the lab and think about it. So I think we experienced what you're saying.
Yeah, and I think it's how we as humans really learn well. We're preparing right now a Duchenne muscular dystrophy masterclass, and it's for general practitioners, physiotherapists, allied healthcare professionals. And I was testing the material with a user just yesterday, and they said, it was your patient and parent pearls video where a parent, they're a set of foster parents to two boys with Duchenne muscular dystrophy, describing their experience. That was really one of the major takeaways for me. While I really appreciated the didactic courses that you offered on the genetics of DMD and the disease-modifying therapies that are about to come to market, it was really those patient stories that I'm walking away from this course. So I thought that was really impactful to hear.
That's amazing. That's exactly what we're looking for. Yeah.
Okay, let's start with one of these questions. Okay, first question for both of you, perhaps. Your proposals are interesting. Have you ever thought of how a disease like Duchenne is confronted or cared for in different countries? They feel that their opinion is it would help actually enrich your work.
Mm-hmm.
I have an example of that. Maybe perhaps I can start. So MDC, together with Canadian researchers, received funding from the International Joint Rare Disease Program. It's a European Joint Rare Disease Program, EJRPD. And we received funding to work together with researchers in Italy and in Germany on a project to look at transition experiences for boys and young men with Duchenne, going from pediatric to adult care. And as part of this project, we've included 6 patient and parent partners in Canada and parent and partners from Italy and Germany as well. And while this is a clinical research example that I'm providing, it's been so eye-opening seeing patients and parents talk about their experiences transitioning from pediatric to adult care, and even seeing the differences of how the different countries include patients in their research teams as well. But it has been quite enriching already in the first phase of this project.
Fantastic. Okay. Yeah, Chris, do you have anything to add?
Yeah, I've actually thought about that and wondered if that would be in line with some of the expertise with colleagues here at our School of Kinesiology and our Muscle Health Research Center, who are more population level research on even patient. How do you put this? Clearly, I'm not an expert. We live and work in North York, Toronto, which is a lot of immigrants from a lot of different countries. And so what I've learned from colleagues with their research in other diseases is that even the uptake of medical advice can differ, whether patients are receptive to what maybe our medical system is prescribing and doing and so on. And again, I'm not an expert, but I think there's a number of colleagues even in our group that would be able to participate in research similar to what Dr. Osman's talking about, not just in other countries, but what they're bringing with them here in terms of perceptions and practices and comfort levels with the health, going through diagnostic procedures. So a bit tangential to this, but those are the. I think it's a great question being asked, and it's not our area of research, but I think that must be done, and I've seen that from my colleagues.
Yeah, really great point. Thanks. All right. Our next question, do you foresee patient engagement becoming required or encouraged in future grant proposals? And if so, how does a preclinical lab go about finding an industry partner to be able to reach those patient populations?
Mm-hmm. Yeah. Would you like to go, Dr. Osman, or would you like me to?
Go ahead.
Yeah. Okay. I think if it's the speed at which it's picking up at clinical grant panels, I mean, I think it's only a matter of time. I've certainly not heard any speaking, I've not read any memos, announcements, or anything that it's coming, but it's inevitable. And I think that's, it's a new challenge and a good challenge for preclinical researchers to think this way. So The sooner we are thinking this way, the sooner we'll be prepared for that. But I also don't want that to be the primary motivation. Like, I truly believe we're going to improve the primary outcome designs of our preclinical research by engaging this process, whether or not the grant panels require it. But yeah, I do think it's also practical. It's going to be coming.
Yeah, definitely agree. I would say, I'm thinking back in terms of the sex and gender training that was first offered by CIHR, and now sex and gender analysis is included in the research grants that are in the research applications through CIHR funding opportunities. So I do think that over time it will be something that will be required. And in terms of how industry partners or researchers can go about reaching out to a patient advocacy organization, a patient organization like a Muscular Dystrophy Canada could be a great way to connect with the patient community and patient research partners. And sometimes it could even be organically as well, as if it in clinical research, a participant can then be returned on to come back and serve as a partner, or organically in different events like the walk and roll that Dr. Perry mentioned, community events as well. But it is nice to see this being included, because I think it will encourage people to think about it at the time of writing a grant. We are currently have an open call for clinical and preclinical research projects. And we've now said we encourage that you include patient and parent partners in your funding proposal. And so this is the first year we've done that. We haven't mandated it, but we look forward to seeing how researchers include that in their proposal.
Yeah, I think some may think that the conversation that you guys are having today is somewhat early, but as a matter of fact, I think it's perfect timing, because if this does become required, this is the time for people to start looking out into who they could be connecting with. And then if this does become a requirement, they already are ahead of the game. So
I agree with that.
yeah. Okay, we have another question directed specifically to your research, Dr. Perry. Mitochondria seem to be affected by many physiological conditions, including muscular dystrophy, aging, cancer, etc. Does that suggest it's difficult to define mitochondria as a mechanism? Would it be rather a response to stress? What are your thoughts?
Yeah, that's a fantastic question. We just published a review article last year that was sort of a hybrid perspective on just because a mitochondrial function may change doesn't mean it's a bad thing. It could have been a stress response. And what do we mean by that? If there was an underlying metabolic problem, and let's put that a little more simply, let's say the cell isn't producing enough energy or supplying enough energy for muscle contraction. Well, mitochondria are very plastic, and what we mean by that is they're very adaptable. They're going to sense a metabolic problem, and they're going to change. And the intent is usually to change in a way that helps the cell fix that energy problem. Now, when you go and measure that response at the same time as you're measuring muscle weakness, it might be tempting to think, oh, well, at the time of muscle weakness, I actually saw this mitochondrial pathway change. Therefore, my hypothesis is it was causal. But it actually could be the exact opposite. It could actually be that, no, that change was actually an intention. And we have a review article on that last year. Catherine Bellissimo is the first author, in American Journal of Physiology, Cell Physiology 2022, for those that want to look it up. And so how do we go about designing studies to establish causality? And I think that's very important. Another benefit of partnering with industry, if you build those relationships, is the compounds they have are not just potential therapies to explore at a preclinical level to decide whether it should go to clinical trial. It's also the exact experimental tool you need to establish causality. So if we add a drug that changes a pathway in the mitochondria and it improves muscle force, I think we just established causality. That mitochondrial response was a mechanism of muscle weakness. But if we add the drug and we do successfully change the mitochondria in a particular way, and there's absolutely no effect on muscle force, well, that's interesting. I wouldn't call that a mechanism now. And that becomes a basic science question of why did the mitochondria adapt and why isn't it affecting force? So the very tools we're using to develop therapies are also the very compounds we're using to develop therapies are also the tools to answer your question. And the last point I'll leave is I do think you're asking the question that's missing in a lot of mitochondrial studies is just because mitochondria change in a disease doesn't mean it was a bad change. It might have been trying to rewire metabolism. Yeah, stop there.
Okay, very clear. Thank you so much. We are almost out of time, but I do want to just squeeze one very last question in. What specific opportunities does a program like this provide trainees, and do you see it benefiting their learning away from the bench? I think this is an important question, so I just want to squeeze that in the last minute that we have.
The program at Muscular Dystrophy Canada, or just the greater idea, perhaps?
Yeah,
Younger?
both.
Um,
I
and
think
and
the
it was,
integration probably.
yeah. So what was could you say it again? Sorry, Sydney.
Yeah, what specific opportunities does this provide trainees, and how do you see it benefiting their learning away from the bench?
Oh, absolutely. First of all, it can benefit them at the bench as well. And I didn't really say that. They become inspired. And I've seen that when I already mentioned to Dr. Osman, we saw a patient with mitochondrial disease talk about their daily life. All the students were just quiet, and I was quiet. And we talked after, literally almost in whispers, like, can you believe what that was like to hear that? We've never heard that. We went back more inspired. So it benefits our bench research. But you're asking a critical question about outside the bench. I think it also builds the students' networks with not-for-profit agencies. I mean, to work with Dr. Osman, I wish I got to do that as a trainee, to see how a not-for-profit works, to see how an advocacy foundation works, and not just as a potential career, but also shaping what is important to the trainee in getting something out of a career. Maybe they will see, wow, as much as I like research, hopefully, I also want to be a little more at the front lines, and they may never have known of this opportunity to be on the front lines working with people with these diseases. And so it exposes them to new career directions, but in a way that they can find something meaningful to guide that thinking. Um, and um, and also, lastly, it's a transferable skill for a trainee to develop communication skills. Like if trainees are most likely going to be creating, 5 things to know about muscular dystrophy pamphlets that can be shared within their muscular disease community. Well, that means our trainees have to actually sit down and think, how do I take complex knowledge and cater it to the target audience that has this lived experience, and maybe I need to contextualize some facts this way. I can't think of a single career that doesn't require that skill.
Absolutely.
Yeah, definitely agree. And obviously, your lab knows what this disease is that they're studying, but to actually hear someone firsthand and meet them who is living with it, I think absolutely you're right. It's probably so inspiring.
Yeah,
It can probably be hard to um, associate, I suppose, when you're studying one specific element of something in a lab, you know, whatever, whether it's in vivo, in vitro, but then to actually, like I said, meet the person living with it and who you're trying to help. It's just, yeah, probably amazing.
absolutely, absolutely. And
Great.
I see a number of technical questions, and everyone asking technical question, you're welcome to email me. My email will be on my website in the handouts. Happy to respond to your emails.
Yeah, let me pin that now for everybody. There we go. Yeah, absolutely. We'll also send all the questions in to both Dr. Perry and Dr. Osman, and hopefully we can get you a Q&A report as well. Um,
Great.
yeah, thank you both so much. It was such a pleasure to have you join, and just really amazing conversation.
Thank you for having me. Thank you. I've learned a lot listening.
Thank you so much also. This has been a great conversation, and I really appreciate working with Dr. Perry and the students and learning from your lab. So I appreciate this opportunity. Thanks, everyone. Thanks for listening.
Great. Yeah, and I have to thank you both for the work you're doing as well. So fantastic. Okay. In closing, I hope that you enjoyed this Inside Scientific webinar produced in partnership with the American Physiological Society and sponsored by Aurora Scientific, and I hope you all enjoy the rest of your day.